You all remember me?
Yeah.
Okay, so my name's Matthew again. I'm from the State College of Florida, right? You all did a DNA fingerprinting lab last week, right?
Right.
How was that? Worked pretty well, right? Okay. So today we're doing another lab, right? And to help me do it, we've got Adrian, Tom, and Brian here right now. Yeah. So they're going to kind of partner up with you guys like we did before and help you do the different steps in the lab. So this lab is called, we're calling it a pGLO. The P refers to a little ring of DNA called a plasmid. And we call it GLOW because that plasmid has been genetically engineered to have a gene, a little piece of DNA from a jellyfish. The jellyfish glows. You're going to take that plasmid that has the jellyfish gene and put it into living bacteria. And guess what? The living bacteria are going to glow. How cool is that? You won't see them glow today, but by tomorrow, if you do it right, you'll see them glow. So let's think about it. If we have this little ring of DNA called a plasmid, but that's just DNA, right? And so you can imagine right here, it has a little piece of, well, not right there, like right here, because it's not calibrated, right on the circle there. It has a little piece of DNA from the jellyfish, right, that tells it how to glow. But there's other little bits of DNA on here too. There's one little bit of DNA that tells it how to make a protein that allows it to live in ampicillin. Do y'all know what ampicillin is?
No.
It's an antibiotic. What do antibiotics do?
Kill.
They kill bacteria, right? So if you just took regular old bacteria and you put them on ampicillin, they're going to die, right? Like you take an antibiotic if you have a bacterial infection, that gets rid of the bacteria. So in this case, if you took these living bacteria and just put them in regular ampicillin, you'd expect them to die. But if they have this little super ring of DNA here, they have a little piece of DNA that basically tells them how to live in poison. Isn't that cool? Yeah. So they've got a little piece of DNA that tells them how to live in ampicillin, a little piece of DNA that tells them how to glow, and the little piece of DNA that tells them how to glow has a little switch attached to it, like an on-off switch. So they don't just automatically glow, there's this switch. The way the switch gets turned on is that you put them in the presence of a sugar called arabinose. If they're in the presence of arabinose, the little switch gets turned on and they glow. But if they don't have the arabinose, then they won't glow, even though they have the switch even though they have the DNA to be able to glow, the switch won't be turned on. Can you all kind of follow that? Yeah. So you're going to have two tubes of bacteria here that you're going to make, right? And one tube you're going to label plus, the other one is minus. So there's going to be bacteria here and bacteria here. The difference is that these bacteria are going to get the DNA, and these bacteria are going to get nothing. Then you've got four little plates where you're going to plate them out. You've got one that's labeled LB. LB is luria broth. That's the food. Obviously, without the food, nothing's going to grow, right? They need to be able to eat. Then you've got two plates that are labeled LB amp. What do you think the amp stands for?
Ampicillin.
Ampicillin, right? So what's ampicillin again?
Antibacterial.
It's the antibiotic that's going to potentially kill them. And then you've got one that's got LB, one that's it's got AMP, and it's also got ERA, it says, which is for arabinose. What's arabinose again? That's the... No, LB is the food. It's got the sugar. What does the sugar do?
Turn switch on.
It turns the switch on. So you're going to put some of the bacteria that didn't get any of the DNA, so the negative ones, right? You're going to put the negative here, and you're going to put the negative here. And the positives, you're going to put the positive here, and you're also going to put the positive here. And then we're going to pop them into the incubator so that they can grow a little bit overnight. What do you think is going to happen here? So these are bacteria that didn't get the plasmid DNA, and they're just growing on regular old food. Are they going to live or die?
Die.
Oh, they die. They're on LB as food.
Oh, yeah.
They're going to be happy as could be, right? They're going to be in there. You're going to have a little lawn. You won't even see individual colonies. It'll be a whole little lawn. It's just happy bacteria living on food. How about the guys that didn't get the DNA that are growing on food plus poison? What's going to happen to them? They're going to die, you would expect. There shouldn't be any little colonies on here at all because they don't have the DNA that would tell them how to live in the ampicillin. How about the bacteria that are here? These now got the special DNA, right? And they're trying to grow on food plus poison. Should they live or die?
Live.
They should live, right? Because they got the special DNA that tells them how to live in this ampicillin. But there you're going to see just little individual dots because only some of them are going to actually take up the DNA. Anybody who didn't take up the DNA, they're going to die, right? Because there's no way they're going to be able to survive in this ampicillin. Do you think these guys will glow?
No.
No.
No. They shouldn't glow. Why won't they glow? They have the DNA that tells them how to glow.
They don't have
Sugar.
the sugar.
But they don't have the sugar that
Turns the switch on.
flips the switch. How about these guys? These guys got the DNA, and they're trying to grow on food plus poison plus sugar. So how's that going to work?
They're going to glow and live.
They're going to live, and anybody who didn't get it would die, so you'll see little individual colonies, and because there's a ravenose there, the little switch should be turned on and they should glow. Are you all following that?
So,
Yeah.
they're going to live, they're going to die, they're going to live, but they're not going to glow, and they're going to glow and live.
You got it. So you
Live,
totally
die,
understand
live,
it.
live.
Now we'll find out tomorrow if it actually worked,
Yeah,
right?
we'll see.
But what you're going to do is set it up today. So the first part of this, you have to get these two tubes. You have to get two tubes, and you all notice you've got two little colored tubes here? And so you're going to label these. What are you going to label these two tubes? Plus and minus, right? And then you're going to take this stuff called TS, transformation. That's what you're doing. This is called a bacterial transformation solution. And you got to get 250 microliters. You're all experts at using these things now, right? These micropipettes.
Yeah.
You got to get 250 microliters into each tube. Basically what this is it's salt water. It's got a little bit of calcium chloride in water. And what that does is it kind of attaches to the membranes of the bacterial cells and it loosens them up a little bit. Yeah? So you've got to get 250 in each one. Can these things measure 250?
No.
No, this one only goes from 20 up to 200, so how could you do that? Oh my God.
225.
Yeah, you could do 200 and then change it and add 50, or if you wanted to not have to change the measurement, you could put what?
125.
Like 125 and put it twice. How about that? Yeah? So you're going to get that into both these tubes. Then you're going to get some bacteria, and this is what we'll kind of help you with a little bit. You've got a starter plate here. This has just regular old plain bacteria with no special DNA at all. You're going to take these loops, and you realize the tip of this loop is sterile, right? So you don't want to touch this. You're just going to pull it out by the end, take a little bacteria, stick it in the tube, and squish it like that and get it all off into that transformation solution. Then you'll throw this away. We're going to keep everything that touches bacteria in these red cups, and then we're going to put it in a biohazard bag at the end, right? Because you're working with a living organism now, not like last week. Then you're going to take another tube, take a little colony, swish, and so you'll have the bacteria in there. And you got instructions there. Anybody know what we're going to do now? We've got two tubes that are identical at this point. What do you think we're going to do now? They're labeled plus and minus. And then what are we going to do to the plus one?
Put the glowing.
We're going to put the glowing, living antibiotic DNA in there. And so we've got it in little tiny tubes sitting on ice back there. We're going to kind of walk around with it. And you're going to put 10 microliters of the DNA into the plus tube. So you need to set your P20 on 10. But the problem with this is, if you just do that, the DNA won't go into the cells. So what you're going to do is they have to sit on ice for about 10 minutes and get good and cold. So they're sitting there shivering like this in their little salty water. The DNA is floating around out here, sitting there, sitting there, sitting there. And then you're going to walk them over to that water bath. And this is where it's very important. You have to have something so you can time it. You all have something you can time it, like your phone or something that has a stopwatch? And what you're going to do, you're going to carry the whole thing, ice and everything, so that they stay nice and cold. Don't just like pull this out and like walk over there and stand around for a couple minutes. It has to be nice and cold like this, and then you're going to dunk them in the hot water for exactly 50 seconds. And what they're going to do is they're going to go like this, and the DNA is going to go in, and then after exactly 50 seconds, you're going to pull them out, throw them back in the ice, and they'll close up again so that the DNA stays inside. That's the only part of this thing that's hard. If you don't do that exactly right, it won't work. They won't take the DNA in. So you're going to do it with both tubes. How long in the ice? 10 minutes. And then you walk over there with the whole little ice bath, and then into the hot water for how long?
50 seconds.
Exactly 50 seconds, and then back in the ice, and then they have to stay in the ice for at least 2 minutes to make sure that the DNA stays inside. Then what you're going to do, because they're going to be massively stressed out when you do all that, right? So what you're going to do is you're going to take this other tube that says LB. What's LB again?
The food.
The food. And you're going to put 250 of this into each tube, and you're going to let them sit there and relax for a little while and eat and be happy, right? And then you're going to take them and put them onto the little plates. Yeah? Yeah. Can you all picture what you're doing? We're going to kind of be around helping. So you can go ahead and label one tube plus, one tube minus, get 250 microliters of the transformation solution in each one, and then we'll help you get bacteria in each one.